I have just created the world's first working time machine! Oh no, I'm late for class! Oh no, my creation didn't send Mrs. Kelly back in time, but it de-aged her, and she needs to teach a class next period. Mrs. Kelly, do you remember anything about cell growth?
Cell growth? Like this?
No, not that type of cell. This type of cell. The cells that make up your body.
Oh, I know. Those type of cells contain a nucleus, a mitochondria, cytoplasm.
I know, but do you know anything about cell growth? Don't worry, I'll teach you. So, first of all, cell growth means two things. A cell grows in size and it copies itself.
Wow, like a printer?
Sort of, except unlike a piece of paper, the cell doesn't need another object like a printer in order to copy itself. It grows and it copies every organelle within it. After that, the organelles move to different sides of the cell and the cell breaks off into two new cells. These cells are called daughter cells.
They're girls? Are there son cells as well?
Well, no. Cells don't have genders. They are just called daughter cells because they come from a single cell, the parent cell. Now, let's get into the inner stages of cell growth. I have a pie.
You have a pie? What flavor? Banana cream? Lemon meringue? Can I have my slice a la mode?
No, you didn't let me finish. I have a pie chart that represents the steps of interphase. This is before mitosis happens, but I will talk more about that stage later. Interphase is basically the steps of cell growth. So we first have the G1 phase. All cells start off in this stage. G stands for growth. In the G1 phase, the cell starts to make proteins that are needed for the next stage, synthesis. Proteins are molecules that are made up of certain types of molecules called amino acids. Remember that an atom is the smallest thing there is. They make up everything, just like how Legos make up Lego castles. A group of atoms that are connected to each other is called a molecule. Amino acids are certain types of molecules that figure out how the protein will work. Proteins are what make up most of the cell. There is also a stage after G1 that some cells are stuck in, called GO. In GO, the cell does not go on to the next step. It is still alive, but it is not prepared for cell division or to be copied. Some cells that are stuck in GO are skeletal cells, or cells in your bones. They never receive the signal or a phone call to move on to the next stage. Cells that can move on to the next step and are not stuck in GO must go through a G1 checkpoint or restriction point. During this checkpoint, the cell checks that it is at the correct size, has the correct material, checks its growth pattern, and checks for DNA damage through proteins. It also checks if the proteins cyclin and CDK are present. CDK, or cyclin-dependent kinase, needs to bind to cyclin. When cyclin and CDKs bind or come together, the resulting molecule MPF calls the cell and tells it that it is ready to move on to the next stage. If the cell finds something wrong or doesn't understand the call, everything comes to a stop and the cell dies or goes through apoptosis. This is kind of like a report card. It checks if you understand all the material in class to move on to the next marking period or the next year. If you fail and don't understand the tests, you are not able to pass on to the next year.
What's DNA again?
DNA stands for deoxyribonucleic acid. It is found in the nucleus, the brain of the cell. Now, all DNA have the same bones, but they have different types of bases or insides. DNA is like a cantaloupe. The skeleton protects the bases inside, just like how the tough skin protects the fruit in the cantaloupe. The backbone is made up of a sugar or a monosaccharide called deoxyribose and a phosphate group, a molecule made of 4 oxygen atoms and 1 phosphorus atom. Now, the bases all have the atom nitrogen in them, but are made up of other atoms as well. We have some that have 1 ring, called pyrimidines, and some that have 2 rings, called purines. The shape depends on how many atoms are in the base. Pyrimidines are thymine and cytosine. One special pyrimidine is called uracil, but we will talk about that later. Purines are adenine and guanine. Now the different bases affect how the DNA will act. Something special about DNA is that thymine will only be with adenine and cytosine with guanine. They are like soulmates. They always go together. Just like how parents are together through the bond of marriage, the bases are together through hydrogen bonding. If adenine goes with cytosine, they will not get along and there will be a problem. The two strands run anti-parallel, with a 5-phosphate on one end and a 3-hydroxyl on the other. Hydroxyl is a type of amino acid. One strand is right side up and the other is upside down. They are joined together in a double helix, which is basically two spirals.
What does this have to do with cell growth? Also, why do we need DNA?
So the next part involves DNA synthesis, basically making two strands or pieces of DNA into 4. DNA is like a blueprint for the cell, and it tells the cell how to work. These different bases are a genetic alphabet that writes instructions for the body. The next stage, S, stands for synthesis. First, there is an enzyme called helicase that unwinds the DNA like a zipper. An enzyme is a protein that wants to make other molecules bond faster. What is going to happen is a new strand is going to bond with an old strand. The old strand is called the parent strand and the new strand is called the daughter strand. This is called a semi-conservative process. Now, there are many origins of replication where the DNA is unzipped and new DNA is created. As they become unzipped and add new strands, the close parts will disappear, creating 4 strands. Different enzymes help build the new strands of DNA. The enzyme SSB prevents the DNA from zipping back up. RNA primase sets the stage for making the new strand, as it builds a segment or wall that won't stay there forever, called RNA primer. This is where uracil comes in. Primase does not use thymine, so adenine bonds with uracil for a short amount of time during this step. The primer will not be part of the final daughter strand. Polymerase comes in and builds the actual strand of DNA from the primer. Polymerase needs a starting point to build off of, so it uses the primer wall. It is like how you cannot build a Lego castle without a base. Because polymerase only works from 5 to 3, the leading strand is the one from 5 to 3, and the lagging strand is the one from 3 to 5. Some parts of the strand lag or fall behind because polymerase bumps into the primer while it tries to work. As the primer is blocking the way, the enzyme leaves and goes to add parts of DNA somewhere else. These parts are called Okazaki fragments. The leading strand, however, is continuous or doesn't stop. The enzyme RNase H gets rid of the primer, and another polymerase enzyme fills in the gaps with the final parts of DNA. Finally, ligase seals the fragments like glue, making the strands continuous. Now we have 4 strands of semi-conservative DNA. Now comes a histone, a protein that is responsible for packing DNA. We must pack the DNA into the nucleus during interphase. The protein and DNA package is called a chromatin. Most chromatin is loosely packed, called euchromatin. It tightens up before we move on to mitosis. Next is the phase G2.
Do plant cells do this too?
Yes, the steps get a little different for plants when it comes to the last stages of mitosis, but we will explore that later. Now let's get into the G2 phase. This is the last stage before mitosis. This part is when organelles called centrosomes form in the cytoplasm. They form with centriole pairs inside. They are of course copied along with other organelles. The centrosomes work sort of like spools of thread or string during mitosis. Before we move on to mitosis, there is one last checkpoint in interphase. This is where proteins check if the S phase was completed correctly and if centrosomes were formed. Now, mitosis.
Hey, don't the steps of mitosis make an acronym? Isn't it PPMAT?
Yes, you are starting to remember information now. I remember you taught us that. It is prophase, prometaphase, metaphase, anaphase, and telophase. Let's start with prophase. So this is when the centrosomes start to produce spindle fibers or strings. This is why I said they are like spools of thread. Now inside the nucleus, The chromatin is packed and stored into shapes called chromosomes, and they look like butterflies. Chromosomes are made up of sister chromatids, which are two blocks of packed chromatin. They look like the wings of a butterfly. The centromere is the part where the chromatids are joined together, forming one chromosome, kind of like the body of a butterfly. In prometaphase, the nucleus's membrane, also called the nuclear envelope, starts to break so that the chromosomes can be free. The centrosome's pieces of string start to stretch and move toward the center of the cell and attach to the chromosomes. They attach to the centromere, and when they attach, the center is then called the kinetochore. The strings that are attached to the centromere are called kinetochore microtubules, and the fibers that are not attached are called non-kinetochore microtubules. In metaphase, the nuclear envelope is completely broken, and the chromosomes are lined up in the middle of the cell in a neat row.
Oh, I think I remember the next part. So anaphase is where the chromosomes get separated into sister chromatids. The strings pull their respective chromatid to opposite sides. Finally, in telophase, information goes to the opposite sides of the cell. I forget what happens next, though.
Very good! Now a band of proteins will be drawn closer together along the middle of the cell, pulling the membrane closer and closer until it pops into two different cells. This part is called cytokinesis. In plant cells, it's a little different. Cell wall parts build up in the center of the plant cells until they are completely separated into two different cells. And then, the cycle happens all over again with the daughter cells. Just remember that sometimes this process can get out of control. There is only a small amount of space that cells can divide in, and when a cell becomes transformed, it means that the cell is dividing even though there is no space. They never die and continue to divide out of control. Sometimes these cells divide so much and so quickly that they don't do it correctly. This is when it becomes cancer.
Gotcha. I think I am ready for my next class. Thank you for helping, Maria.
So that is cellular growth. Any questions?
Where are your parents, little girl?
What do you mean? Oh wait, I'm still 5 years old. Maria!